Many of the genes that we have are ancient. They haven't changed much over billions of years, and they can't change much because to change too much would make life impossible. So for instance, certain genes that code for enzymes that are involved in aerobic respiration haven't changed much. And they can't change much because if you change the most important part of the enzyme, the so-called active zone, it just simply won't work. And it won't fulfill some very important task. But other genes have changed. If you watch the video from Sean Carroll, you've seen that he says that for a lot of cases, evolution involves simply repurposing genes. So taking genes that are already there and those genes somehow change so that they have a new function, a brand new function. But then there is the question of where do all of these genes come from? And so actually the traditional explanation is that they come from cases in which genes have been duplicated. The genes are duplicated, and so you have two working copies of a gene in the organism, and actually one of those copies goes on and changes so that it ultimately does something else. So a mutation that affects it will make it perform some other type of function. More recently, people have thought about different approaches to this, and actually it seems to be the case that there is what's called de novo, brand new genes that pop up from the non-coding sections of DNA. So as you know from the study of DNA, we have the sections of DNA that are protein coding. They code for something. And then we also have non-coding sections, and this non-coding parts either are so-called junk DNA, or they may be involved in the regulation of the genes, but they don't code for any protein. So I just read an article in the New York Times by Carl Zimmer who summarized some research showing that once in a while there'll be a small part of that non-coding DNA that'll convert into a coding region. Here's how that works. All protein coding regions begin with a start sequence. That's how the cell knows it's a coding region. I'm showing this in the illustration here. The cell treats everything after the start sequence as a coding area until it comes to a stop sequence. Mutation can, by chance, introduce a start sequence into the non-coding region. If that happens, the cell will then treat the region after the new start sequence as a coding region. So it's as if the cell created a new gene, and in fact it has. So what type of protein will result from this new gene? Well, it's hard to say since the non-coding region is random junk. It may be that that protein will do nothing, or it may actually be harmful. But it will be there, and once it's there, it's subject to natural selection, just like anything else. So if you want to read further information about it, here's the information.